Welcome to Neomatician webinar series, where academia and industry experts converge to exchange ideas, engage in thought-provoking discussions, and seize networking opportunities. Neomatician offer a diverse range of courses, organize captivating webinars, foster collaboration, and promote continuous learning and knowledge sharing. Our aim is to cultivate the next generation of technologists and innovators.
Good morning, afternoon, and evening, esteemed members of Neomatician community and all life science enthusiasts from across the globe. I am Dr. Priti Prasad, Program Manager at Neomatician. Thrilled to welcome you to yet another insightful sessions of our webinar series. Today, we are virtually gathered for the webinar entitled Pharmacogenomics, Unveiling Personalized Medicines. We have Dr. Manmohan Pandey with us, a genome informatics expert having more than 12 years of research experiences in variance analysis and clinical genetics. He obtained his PhD from the Amity University, India, and currently serving as a Senior Bioinformatics Manager at Agilus Diagnostic, India. Without further delay, I am delighted to pass the stage to our esteemed speaker, Dr. Manmohan Pandey, who will walk us through the intricate world of pharmacogenomics.
So, good evening, everyone. So, I'm going to take you on the tour of pharmacogenomics, where I will cover different aspects of pharmacogenomics, like what is pharmacogenomics, what are the different dosing guidelines, then some case study, and then what is the use of pharmacogenomics. So, I will cover all these things in details. So, before going into the details of pharmacogenomics,
Dr. Pandey, sorry to interrupt you. Please, if possible, you can start your video so that we can visit, we can see you as a face-to-face.
It is showing that you cannot start your video because the host has stopped it.
Yeah, okay. Let me do it.
Okay. Start.
Yeah, Dr. Pandey, continue.
Now it is visible. Let me check. Okay, fine. So before going into the details of pharmacogenomics, first let's understand what is pharmacogenomics. So normally in developing countries, around top 10 cause of disease and morbidity is adverse drug reaction. Normally we complained about that we have there's many drugs that doesn't suits us. So and also the problem is with the different drugs also. So pharmacogenomics is the answer to all these things. Around 10-20 to as per one study, around 20 to 60% of the individuals doesn't normally one or two drugs doesn't suits to them. So this is where the pharmacogenomics comes into the picture, where the genetic makeup of an individual or the variation in your genes will affect your body response to the medication. Now here we have to understand two thumb rules. The first is that any disease occurs because of either accumulation of some products or deficiency of some product. Secondly, if any variation exists in the enzymes or protein, so it may either enhance the activity of the protein or it may decrease or halt the activity of the protein. Normally, in case of normally we have for any drug, ADMET property is followed. That is how the drug is metabolized, how the drug is absorbed, distributed, metabolized, and excreted. So if any of these enzymes, so all those enzymes which are responsible for the metabolism, absorption, distribution, and processing, and finally the breakdown, if any variation occurs, so these variation can have a significant difference in our response to different drugs. So normally what at present level we have drugs designed at the population level. That is one drug fits to all. But in case of pharmacogenomics, when you're going for the pharmacogenomics, so it holds the potential for an individual drug, for a drug at individual level, at individual precision level, as per the genetic makeup of the individual. Now with pharmacogenomics is divided into two parts: pharmacokinetics and pharmacodynamics. So pharmacokinetics is how the drug is distributed in the body. That is absorption, distribution, metabolism, and excretion of the drug. And second is how this, what is the body's response to these drugs. So that is called pharmacodynamics. So what, whether that it increase the efficacy of a protein or it decrease the toxicity in the body. So all these comes into the pharmacodynamics. Now, we have different genes, as I've already told that there are different genes, like different genes are there, which acts on the drugs. So these normally the cytochrome P50 genes is majorly the part of different pharmacogenomics drugs. And if any variation exists in these drugs, so it will either it will either inactivate the protein, or it will enhance the activity of the protein. So normally around 99% of individual, according to one study, has one PGX version. So this is the beauty of PGX. Like if that we have to consider pharmacogenomics variation, and based on that we have to take, based on that different dosing guidelines are there, different alternative drugs are there. So since every individuals have at least one PGX variation, so it helps us to find out which variation will cause what type of adverse drug effect, or what type of drugs are suitable for us. Normally, these drugs are categorized into antidepressant, heart disease medication, painkillers, etc. As per one study, like one of the recent study by an Indian scientist, each individual carry 8 PGX variants. So this is highly recommended in case of Indian, because we are taking drugs based on one population, one size fits to all, and it will affect the adverse drug reaction level, right? So normally, in case of traditional medicine versus precision medicine, it is normally all the drugs are prepared for one size that fits to all. That is, it is based on different. It is based on different. It is based on single population, right? So it is based on all population. So there is a trial and error approach, and it must have ADR or side effects are there. But when we are going for precision medicine, so in that case we have minimum side effects. It helps us to assign the optimal drug type and doses. Now, normally what happens, suppose I'm taking an example of an enzyme, which is metabolizing enzymes. So suppose a drug is there. So if any variation exists, so that drug either enhances the activity of a protein. So that is called ultra rapid metabolizer. And if it decreases the activity of the protein, so it is either intermediate metabolizer or poor metabolizer, right? So if the metabolism is very swift, the activity of the metabolism is very high, then it is ultra rapid metabolizer. And if it is normal, so it is called extensive metabolizer. So what happens that in case of ultra rapid metabolizer, the drug is metabolized very swiftly from the body, so we need a high dose drug. And in case of poor metabolizer, the drug is retained in the body, so that may cause toxicity or accumulation in the body. So that requires a low drug. So this is what the basic behind the pharmacogenomics approach. Now, if any drug is there, so if we are going for pharmacogenomic testing, so we can have either a good response to that drug, or we can do the doses adjustment. That is either because of that pharmacogenomics variant, either that doses will be needed in high dose or in low dose, or or alternative drug can be prescribed. So based on this, we can prescribe different drugs. We can see what is the effect of different drugs on an individual. So what is the benefit of pharmacogenomic panel to healthcare providers? So as I've already told that since we can have those variants which may hamper or increase the activity of protein, and based on that we can recommend which drug or what is the doses of the drug. So the minimum trial, it will minimize the trial error. Normally when we go for any drug, so if it doesn't suits you, so the doctor recommends some certain other drug. So this is trial and error approach. But these trial and error approach can be minimized. We can early identify the adverse drug reaction, and doses adjustment, precision dosing can be done. This because of this, whatever adverse drug reaction is there, so for that we have minimum hospital stay is there, and accurate drug selection is there. Personalized medicine also has a powerful, it also holds a potential in drug discovery based on individual genetic makeup, and it has a maximum therapeutic value. Now we have different guidelines, as you know that in rare diseases we have ACMG guideline. Similarly, in case of PGX, we have three or four guidelines are there. That is CPIC guidelines is there, DPWG guidelines is there, CPNDS and RNPGX guidelines are there. So these guidelines are governed by different consortium. That is, one is US based consortium, that is CPIC. Then there is a Dutch consortium, that is DPWG, and there is a Canadian consortium, that is CPNDS, and French consortium, that is RNPGX. Now these are the maximum gene, that is like these are the genes which are covered. We have list of genes, so you can see lots of CYP2 CYP genes are covered. These are metabolizing genes, and these carry around 30 different that can be act on different medicines based on the variation. So this is the comprehensive list. Around 32 genes are covered right now. It is around 200, more than 200 plus genes are covered, but these are the major 59 genes for which we have pharmacogenomics recommendation is there, out of this 32 is most important. Now these are the more than 100 drugs which are included in case of pharmacogenomics. So you can see like these drugs are from general medicine to a different specialty wise, like we have uh, we have nephrology associated, we have neurology associated, we have cardiology associated drugs are included in that. Not only not cardiology, but also oncology associated drugs are there. So these are the specialty which is addressed in pharmacogenomics. So we can like lots of overlapping drugs are there which are which may be there in the specialty. So we can cover different specialty in case of pharmacogenomics, where we can cover different psychology drugs. We can cover in different environmental metabolism, cardiology, dermatology, endocrinology, so lots of drugs can be covered in case of different specialties. So this is like applicable to most of the specialty, and it gives a blueprint of a particular individual, that which drugs will suits to a particular individual uh, to whole of their life. Now, what is the pharmacogenomic analysis pipeline? Normally, the pharmacogenomics, whatever sequencing is done, it is either done through Sanger sequencing. If you want to cover one or two genes, or like 2 to 5 variants, then you can go for Sanger sequencing. But it is very costly, and it is very time taking. Secondly, you can go for there are different microarray tests are available, where you can find out different variations. And thirdly, you can go for either whole exome sequencing or whole genome sequencing. So normally, after sequencing, and when we align, once we get the data, we align to onto the genome. And once we align onto the genome, then after variant calling, we get the variants which are either single nucleotide variants, CNVs are there, then insertion deletion variants are there. So based on those variants, we can have either single variant alleles are there, or we have multiple variant. That is, we can have alleles which are which may be single pharmacogenomics variants are there, or we can have a combination of pharmacogenomics variant, that is multiple variant alleles are there. So based on that, diplotypes are made, and after that we have different annotation methods. Like all these variants are all the known variants, which is based on different studies. I will just show in the later slides how these studies will help us to find out the strong association, or the association of a different variant to certain disease. So based on that, as I've already told, that we have clinical guidelines, and these are well established guidelines in which we CPI. So these are annotated. These variants are annotated. Whatever is present in case of CPIC, DPWG, CPND, and RNPGX. So they have list of variants which are pharmacogenomic active variants, and based on those variants, based on those annotations, we can recommend whether we have to avoid the use of that drug. Based on that recommendation, whether the drug should be avoided, it is used with caution, or we can use with routine use. Now after that, the phenotypic, based on the phenotypic prediction, what is the effect of that drug, whether it is increasing the toxicity, whether it is increasing the efficacy of the protein, whether it is increasing the metabolism of the protein. In that way, the dosage is also decided. So based on this pharmacogenomics analysis pipeline work. Just cover the star alleles. So whatever diplotypes are there, so that is pharmacogenomics testing is based on the star alleles. So star denotes the nomenclature assigned to ADME genes. So star one, that is whenever we are aligning to the genome, so whatever is the default genome, right, what is functional alleles are there, so that is called, that is denoted by star one. So if it is star one, star two slash star two is there, so that means heterozygotes. And other designation which define the haplotypes is star two, star three. So once we have different variations, so the designation can be given like star two, if suppose from A to T variation is there, and default is A, so A is given as star one, and if T, it is if the variation is from A to T, then the star 2 is given to T. If it A to G is there, so it's variation. So in that way, so it is what the haplotype is given. So when I will just go into the last of it, about the reports, so you can see there. Next slide, please.
Yeah, sure.
Now this is a study on Asian population. So you can see that since we have different pharmacogenomics variation, so you can see that the variation which is very which is poor metabolizer in case of CYP2C19, the frequency is very high in East Asian population and southern Eastern Asian population. Similarly, there is a gene called HLA-B1502. So that is also the frequency of this variation is very high in case of Asian population, especially in East Asian and South Asian population. So this means that pharmacogenomics is recommended because it is there in the case of our Asian population also, where we cannot take the drugs which is one size fits to all. Next slide, please.
Yeah, it's moving. Just
Now we can see that we have different drugs, like we have based on our Asian population, we have drugs like clopidogrel is there, which is poor metabolizer in case of Asian population, and you can see around 2x increase is there, like in Asian population, the frequency of poor metabolizing variation is very high in case of East Asian population. And that's why Indians are prescribed for antiplatelet therapy for especially clopidogrel is given in less in case in less doses for Indians. There is also one more drug, that is warfarin, tomaxifon is there. So all these drugs are population specific also. So that's why while considering the drug doses, pharmacogenomics is recommended because it gives us an insight where doses criteria can be adjusted, where we can get into the insight about the doses adjustment, about the different alternate prescription of the drugs. Next slide, please. Now this is just a continuation of the slide. So we have different drugs which are, especially which are these are psychiatric drugs, and this is the role of these drugs in especially in psychotic management, as well as pain management, as well as neurological condition is very high. And this should be given this should be given with caution in case of Indian population or Asian population. Next slide, please. So this is what we have already talked about. So Indians are poor metabolizer of CYP2C19. So there are many drugs are there, like clopidogrel is there, then cetoprorom. These are these are the drugs which are antipsychotic drugs are there. So all these drugs are around 13% of Indians carry these mutations. So the doses must be taken, must be given in caution, especially, but not all the drugs are have some have a problematic effect on Indian population. There are some variation which is also good for in Indian population, and as compared to other population, like there is a variant that is called RS1162 01358, which is found in C8A gene, which is when we compare with other population, it has better treatment outcome for imatinib mesylate, which is a drug used to treat chronic myeloid leukemia. So Indians or Asian population have that variation in which the treatment of they have better opportunity for treatment of chronic myeloid leukemia as compared to African and Asian population. Next, please. So these are the different, like I'm just explaining that these are the different uh, drugs are there, the different variations are there, which is responsible for different drugs. So which must be taken in caution, or which so it is done on a population study, and based on that different recommendation of drugs can be given. So we have oncogenic drugs are also. Okay, next. I've already discussed this. Next. Okay, so I will just discuss how pharmacogenomics works. So I've already given some glimpse of pharmacogenomics working. So normally we have different metabolizing enzymes are there, different we have different adsorbing enzymes are there, different distribution enzymes are there, and different excretion enzymes are there. So this is the working of a metabolizing enzymes. So suppose if the variation is if there is no variation of if or if the variation doesn't cause much variation in the efficacy of the protein, so the normal breakdown will be there. But if the variation increases the efficacy of the protein, then fast breakdown will be there. And for this, since the drug will be metabolized very swiftly from the body, so we need more and more doses for that, uh, in order to have a proper response of that drug. Normally, if the slowdown of the drug breakdown is there, that is if the drug is poor metabolizer or intermediate metabolizer, so we need less drug, because because if the metabolism won't be there, so it will accumulate in the body, and it will and that will cause toxicity in the body, so we need less drug. Next, please. So there is a drug called amitriptyline, for which we have two genes, CYP2D16 and CYP2C19. So if any variation is there which makes the drug as a which either halts the activity of protein, or it decreases the activity of protein, so it will be poor metabolizer, and we need less doses. Similarly, if the variation enhance the activity of the protein, then we need then it is ultra rapid metabolizer, and we need higher doses. So normally amitriptyline is a tricyclic antidepressant which is used in the treatment of several psychiatric disorder, including major depression, and it is one of the highly known drug in case of several psychiatric disorders. Next, please. Now there is one variant of this also, that is clopidogrel. That is it is a drug which is used as a blood thinner, and this drug is in case since it is in the inactive form, and when it is metabolized, so it is in active form. So once it is in active form, so it just like in other drugs, if the metabolism is very slow, if it is poor metabolism, so we give less drug, and if it is ultra rapid metabolism, we give more drugs. But in this case, it is totally antagonistic, in which if poor ultra rapid metabolism will be there, we give less dose, and if poor metabolism is there, we give high dose. It is because the clopidogrel is an inactive product, which after metabolism it turns into active drug. So once it is active drug, so if the metabolism is very high, so it will be like it will turn into active drug, and it will have more it will be deposited more into the body and accumulated in the body, and it may cause some ADR. But if the metabolism is very poor, so it is not it is not translated into active metabolite, and then we need more doses for this. Next, please. Next slide.
Yeah.
Huh. Now we have a drug receptor. We have like if any variation is for drug distribution, we have drug receptors. So this is very classical example in which some breast cancer makes uh, HER2 receptor. So if any variation occurs in any individual, and if this HER2 receptor is not there, or very less HER2 receptor is there, so we cannot use an anti-cancerous drug, that is TDM1, for this, for killing the cancerous cell. So for this, uh, we first find out whether there is any mutation. If the HER2 receptor is there, if there is no mutation in the HER2 receptor, so TDM can be used. So this is also a classical example of pharmacogenomics. Next, please. Now, for drug uptake, like whatever drug is absorbed, so if the normal uptake will be there, so drug will be work as expected. But if there is a very decreased uptake is there, so it will so if decreased uptake will be there, so it will be accumulated in the body. And if increased uptake will be there, so it will be processed swiftly. So because there are cardiological drugs called statin, which lowers the cholesterol. So it is transported by a protein called SLCO1B1 gene. So if there is any changes in the gene, and if because of that, if the uptake of the drug is slowed down, so because of that, there is if that variation is there, which decreases the uptake of uptake of that drugs, so there is a chance that we need to increase the drug doses. So next slide, please. Now there is if it is poor absorption is there, we need higher doses, and if rapid absorption is there, we need less doses. So this is what the criteria for pharmacogenomics. So in case of higher doses, semestine can build up in the blood, causing muscle problem, including weakness and pain. Next, please. Now for clinical, we have different level of evidences. Like in case of normal, if we are saying that we have if uh, for any evidence, like what is the pharmacogenomic evidence, that whether we should consider what is the strength of association of certain drug evidence is there, whether we should follow that guidelines or not. So that is the clinical annotation level of any evidence. So if the clinical level of annotation of evidence is very high, so that dosing criteria is recommended. But if it is very low, so it depends upon the patient phenotype, patient uh, uh, it depends upon the patient, how the patient is reacting to that particular drug. If the drug is, if they are not they are not reacting good to that drug, then that might be recommended. But level 1 and 2 has strong association, and that can be recommended. Next slide, please. So I'm just giving you the details of level 1 and 2. So if high level of evidence is there, like 1A 1B, so it is because it is done on very large cohort. And that is it is done by different people, and they have done on large cohort. And it can be uh, preferably has strong effect size and replicated in different studies. So that is categorized as 1A and 1B. So if any variation which is falling in 1A and 1B, and whatever drug association is there in case of 1A and 1B in CPIC, so that is highly recommended, and that dosing guidelines can be taken further. But if it is in and if it is done on a very small on a very small set, that is within a VIP, very important pharmacogene, but still the cohort is large, then it is called 2 and 2, that it is called moderately high. But for third and fourth, it is normally not recommended, and it is based on the person's phenotype. Next, please. Now, in case of our pharmacogenomics panel, we have covered around 206 genes. The total number of genes associated with clinical annotation dosing guidelines, as I've already told, it is 32, and the total number of drug is associated with 609. So, around 250 drugs are associated with different dosing guidelines. Next, please. Now, this is a glimpse of a report, how a pharmacogenomics report is done. So once we do the analysis part, so once we are doing the analysis, so once we have those variants which correlate with the variants which are there in CPIC, DPWG, or RNPDX. So based on that, whatever recommendation it is giving, so based on that, it is giving whether it is giving whatever dosing guidelines it is giving. So if we have to change the alternative drug for this, or it is causing a very high ADR, alternative that is if it is causing very high ADR, so we have to avoid use. So there are three categories. in case of whatever report we are generating. The first category is to avoid use, that is we have to consider alternate medication for that. Next slide please. Second category is use with caution, and third category is routine use. So whatever will be the variation is there, and based on that, whatever study it is given in CPIC guidelines, so based on that, the report is categorized, the drugs is categorized for a particular one. So it differ from individual to individual based on their variation. So any individual who may be uh, have a genetic variation with use with caution, the second individual may have routine use for that. So that may be there. So in this way the drugs may be categorized as per the variation. Next please. Now this is recommendation. So we have different recommendation. So you can see that in this case clopidogrel is a drug, and we have genes and diplotype. So these genes and diplotype is based on different variation. So if in this case this is a heterozygote variation is there, so based on that we have different guidelines are there. So every variation, if you are aware of ACMG guidelines, or if you are aware of rare disease disorders, so every variation has some specific value, and every variation has some specific meaning in case of when we are going for ClinVar, whether it is pathogenic likelihood. So similarly is with these guidelines also. So whatever variation is there, which for which we have a very well defined studies are there, that is based on different criteria, the which, stages of which I have already discussed about the association, like whether it falls in 1A 2A category. So based on that we have different guidelines. So in this case you can see that CPIC, this particular variation is there. So CPIC, for this variation we have to use as per CPIC, we have to use some alternative drug, because this may cause adverse drug reaction. Next please. Similarly, I've taken example of citaprolol and tacrolimus. So for this, it should be used with caution, and the drug doses is defined. So in this case, if you talk about tacrolimus, so it is recommended to increase the doses of the. So all these things depends upon what type of variation we are getting, and what is the guidelines for those variations, and it is done on a very well defined studies. Next, please. Now these are the clinical annotation. So clinical, and based on the clinical annotation we have different drugs, like what is the effect of that drug at phenotype level, whether it is causing, as I've already told you in the analysis pipeline, whether it is causing toxicity, whether that variation is increasing the toxicity level or the metabolism level. Based on that the doses will be defined. So if the toxicity level will increase, the doses will be decreased. If the metabolism will be increased, so the doses will be decreased. So based on that these things are done. So these are the clinical annotation. Next please. Next please. Now we have different pharmacogenomic resources. Like we have different clinical pharmacogenomic so these pharmacogenomic resources are uh, we have uh, we have two resources. So one is CPIC, DPWJ, RNPGX. Similarly we have a very collaborative resource that is called PharmaGKB, which is on which you can find out all these mutations. It is same as what we do in case of ClinVar, in case of rare diseases. Next please. Now these are the different features. So what happens that for pharmacogenomics variant analysis we can have different methods. We have Sanger method, we have array based method, we have so since Sanger based method is not widely used, because it is
Dr. Pandey, your voice
limited
is
to we
breaking.
have more than 5,000 variants in case of but uh, okay, now it is fine.
Yeah, it's fine. Yeah.
Okay, so what happened that we have array based method or WES or WGS based method. So this is when we want to cover a very large portion of pharmacogenomics variants, so we can use either of these method. There are some we have some there are some uh, uh, there's some advantages and pros and cons of these methods. So in case of array based methods, the total number of genes, because since it is an array based technology, so the limited number of genes can be there, and that is the beauty of WES and WGS, that we can cover a whole exome where lots of genes can be covered. So all those variations which may be unique to a particular population and we don't have any other studies, so that variation can be also retrieved. So this is good for the research purpose, where we want to find out a particular variation in certain population, and based on that we can give the recommendation. So it is good for all those novel variation which may be there. So this is beauty of WES WGS. And the imputation accuracy of array based method is very low. So what is imputation? That is, it is ethnicity based. So any variation which is like normally all the array based methods are uh, based on either a Caucasian population, African population, or they are based on Australian or then American population. So whatever variation which is there in case of Indian population or Asian population, that is called imputation, and that imputation accuracy is very low. We may miss some of the variant which are there in case of Indian or other population. Now detection of low frequency and rare variants are very low in case of array based method. So every method has some pros and cons. CNV is very good in case of array based method, but in case of WES and WGS, that might be compromised. So all these things, as per like different, we have limited or predefined number of genes. So it totally depends upon your requirement, what exactly you want to go ahead. Because for array based method, it is much cheaper than whole exome or whole genome based method. But this is a blueprint of your whole genome, the blueprint of an individual. So this is not going to change uh, uh, to whole of your life. And whatever drugs which is prescribed to you, whatever drugs are there, whatever variation is there in your genome, based on that you can take you can have a blueprint of your of what drug you have to use in the future. Next please. So these are references. Thank you.
Thank you, Dr. Pandey, for sharing your enlightening insights on pharmacogenomics and its implication. And now it's time to take questions from our participants. The first question is, what is PGX variants?
So PGX variant is it is normal variant, like whatever known genes are there, with the list of genes which I have shown you, like whatever genes I have shown you, 59 genes or around 200 genes are there, which is already well defined. So all those genes which are there as a metabolizing enzymes, which are there as a distribution enzymes, so all those genes which are there, they are if any variation exists in that, and if it is proved on a cohort with which I have to like already shared, that all the variants are based on a different level of confidence, right? So these are the PGX variants.
So another question is that if multiple variants are seen upon whole genome sequencing in several of cytochrome genes in individual's genome at the same time. So the question is that, so how we can see that for PGX guidelines mentioned earlier, as you mentioned earlier in your slides. So we should go for drug dose adjustment in such case.
Yeah, it totally depends upon the, see, the first thing is we have to follow the normal whole exome pipeline, that whether that those variants can be reported or not, like whether those variants have good quality scores or not, whether those variants have better depth or not. Once it is there, so we can have multiple variants together, and based on that we can give the recommendation. So but it should be, variation should be there in CPIC guidelines. This is the mandatory thing, but that variation should be there. It should be the association study should be done with 1A 2A, or with 1 to 2 stage annotation should be there. So that should be there, so we can go ahead with those variants.
Okay. So what is in your opinion on how computational drug discovery can aid in the pharmacogenomics advancement? So there's a correlation between computational drug and the pharmacogenomics advancement.
we can see because if any variation is there that affect at the level of the protein, right, at the protein structure level. Because if we are saying something like, suppose if we are saying that a particular protein is there, and because of because of that variation, if efficacy of the protein is decreased or increased, so that is based on that, if the protein efficacy is decreased, so based on that we can go ahead with the CAD criteria, and we can find out those things. But still, when we are going for computer aided drug designing, um, it is still very limited things are there in that. So but we can go ahead with that.
Okay. So another questions with the integration of genomics and epigenetics, that as you said that with the gene mutations there are different with the changes of gene, they are having changes in the efficacy of drugs. So the question is that role of epigenetics with regards to change in genes behavior that might be disadvantage in mapping PGX. So is there any role for the epigenetics on the gene mutations?
Right now
Uh,
we don't have any finding for epigenetic because it is totally based on the variant analysis, and epigenetic is somewhat different, and it is not still, it's still in the naive stage. Whatever is done on at the variant level only.
Yeah, so might be in upcoming futures, you, we, and as a scientist like Dr. Pandey, we can will work on that. Once we cover the genome sequencing, we will cover the epigenomic sequencing, that how it gets affected with the PGX variants. Yeah, Dr. Pandey?
Yeah.
So next one is why it is difficult to detect CNV using WGS and WES, and with better with array-based methods. So why it's yeah.
See, actually, for WES and WGS method, the main for whole exome sequencing, the problem with CNV analysis is that two things: whatever is there in the exonic region, that is only detected, and it is the intronic region is not detected, right? Because a whole exome is the collection of the exonic region bed file, right? Secondly, the in case of whole exome sequencing, the depth is not uniform. So there is a possibility that because of the ununiform depth, we may especially not in the case of homozygous deletion, but in case of heterozygous deletion, that may be missed. In case of whole genome sequencing, it is better, but CMA is regarded, because since CMA is regarded as one of the best tool for best method for CNV analysis, uh, because all those CNVs which are there in case of which are already known is covered in in CMA. In case of whole genome sequencing, it totally depends upon your depth, like what depth, at what depth you are sequencing the things. And lots of CNVs, especially those CNVs which are structural variations are there, that is problematic in case of CMA also. But still, CMA is regarded as the best method in case of as per WGS and WES.
Yes, and what is a diplotypes, as you
See,
mentioned in your slides? Yeah.
diplotypes is nothing but it is like we have we are taking two alleles at a time. Like suppose there is a deletion called CC is there, and we have a variation from C to A. So we have two alleles at a time, so it will be CA. From CC it converts to CA.
So we have a number of questions in our Q&A option. So yeah, how much depth is required to consider a variance?
Normally it is 20x, but it totally depends upon what type of variation you are looking for. Like if it is a heterozygous variation, then I will recommend greater than 20x, because whatever the alternative variation will be there, that can be seen. But if it is homozygous variation is there, then you can go ahead with 20x. Minimum 20x is there, but upper number is far better for the confidence of that variation.
So for next question is that what is the metabolism is the only cause for variance level of drug tolerance?
No, metabolism is not the only cause, because there are some proteins also. If there is variation, especially the oncogenic proteins are there, we have list of, but that is covered in the drug label annotation. So that is in third or fourth annotation association studies. But
we have, as I've already told you, that apart from metabolism, all those drugs which are receptor based, that is which may be doing adsorption, which may be doing this distribution or excretion, that can be also taken into the picture. But maximum metabolizing enzymes.
So another question is that if there is a 4 variants expected to represent the star allele, so what we have when we have got a lesser than this, then should we
See,
go further?
I think star allele here people are very much confusing. See, star allele means you are saying on a particular position. Now, on any position, only two variant because we are not interested in polygenic variations, right? Polyvariants. Two, if any two variation can, either it can be convert from A to T or A to G or anything will be there. On different positions we have different pharmacogenomics variant, because CYP2, suppose I'm just taking an example of CYP2 gene, 2D6 gene, so it have different variations at different position, and it may that those different variation can may affect the structure of the overall structure of the protein. So it totally depends upon the what variation is there, what sort of variation is there, whether deletion is there, insertion is there, missense variation is there. So what is the collective effort of those variation on the structure of the protein? So based on that, these clinical guidelines are given, and it is based on certain ranking criteria also. And it is not that if we got 4 or 5 variation in a particular pharmacogenome, whether those variation have some impact or not, and whether it is clinically reported or not, that matters.
Okay. So at every condition, we should follow the guidelines.
Right, because in guidelines the variation is already it is already described. So everything only few only like limited variations are there which may cause pharmacogenomics variants, or your dosing guidelines can be changed. It is not that any variation you are looking for, and you can say that these variation is causing a structure a change in the structure of the protein. It is whatever is already done on a very large number of cohort, because that will represent whether any that is giving the association studies, and it will represent the confidence of that variation. So until unless it is not done on the
Yes.
very large cohort, especially for, if you see that fourth category which I have already mentioned in the slide, so that fourth category is not taken as much because it is not done on a very large cohort. Until unless any study is done on large cohort and it is replicated in that cohort, you cannot say that is a pharmacogenomics variation, and you have to just change the doses, or you can have an alternative doses.
Last questions of this webinar, because we have a time constraint. So interested in pharmacogenomics, what education should we take?
The first thing what I see is what type of sequencing platform you are you are doing. Like if you are going for Sanger sequencing, so you may design primer for which variation you want to consider. So suppose if you have highlighted some of the variation which is present in the population, so based on that you can design the primer, and whatever variant you are getting, you can use the databases which is already there, like CPIC is there, Pharma GKB is there. But if you are planning for any whole exome sequencing or whole genome sequencing, because this is also so for the first thing, what is the pipeline for whole exome sequencing or whole genome sequencing? That must be that must be you must have expertise in that. After that, whatever variant, because in case of whole genome or whole exome, in case of whole exome you are getting more than 80,000 variants. So in order to filter those variants, and then you are then you are filtering those variants based on the pharmacogenomic variant, so it needs a good computational skill.
Okay.
Hmm.
So thank you. We have a lots of questions for today's webinar. We will continue these questions on our upcoming webinars. So with this, I would like to thank Dr. Pandey for sharing your experiences and knowledge with us. And thank you all of you for joining us today. So let's continue our journey of exploration and collaboration. Please spread the word and invite others to join our vibrant community. Together, we can make a significant impact in the world of science and research. We look forward to seeing you all in our next webinar, where we will feature another exceptional life science leader. Thank you, Dr. Pandey. If you have any feedback with us, you can share.
Thank you everyone, and sorry for the interruption actually.
Yeah, it's okay, Dr. Pandey. Yeah. So, okay. Okay, thank you all.